Now it's my pleasure to introduce Dr. Alice Hayward. She is a plant molecular physiologist leading a group focused on tissue culture innovations for plant propagation and conservation at the Queensland Alliance for Agriculture and Food Innovation at the University of Queensland. And this morning her presentation is on in vitro biotechnologies for Myrtaceae on the brink, progress and possibilities. It's my pleasure to welcome Alice. Thank you.
Hi everyone, thank you so much for having me. This is the first time I've been able to present our work to the plant conservation community. Traditionally we've worked on horticultural species for mass propagation for industry, but now we're very passionate to contribute to plant conservation for rare and endangered plants of all kinds. I'm working with a few species, including with Denise on the macadamias as well. But today I'm talking about our work on Myrtaceae. So we're very fortunate. My boss, Centre Director, who has now left UQ, but left an amazing legacy, was able to establish this lab here at UQ. Her name was Nina Mitter, and we are very lucky to have the entire top level of this $20 million refurb building, where we can have 23 laminar flows. We've got 6 independently controlled growth rooms, a lot of controlled environment cabinets, and we're really here to build capacity in vitro technologies for plant species. Our lab traditionally has worked across a few areas, some genetics or molecular physiology of plant development and responses to environmental stress, but also propagation. So we're really focused on tissue culture as a propagation tool, also in vitro breeding, getting into gene editing and those kind of things for horticulture, and germplasm banking, which is what I'm going to be talking about today. So I like to put tissue culture at the middle of everything because I'm very biased. But what we really focus on is elite plant supply, gene editing, as I mentioned, germplasm banking, but it's also a really useful tool for just understanding plant physiology, the role of hormones, plant development, little things that are newly coming to light, like the role of microRNAs in plant signaling, those kind of things. We can explore those in tissue culture. And also cellular agriculture, so that's becoming important as people try and more sustainably produce proteins and pigments and those kind of things. So what are we doing in ex situ conservation? You guys would all know there's a few options for conservation of plant species, in situ and ex situ. Ideally, in a beautiful, perfect world, we'd have all of our plants beautifully maintained where they're supposed to be with no threats or impacts. Obviously, that's not possible. So what we're working on is ex situ conservation. There's a few options for this. There's field banking and also seed banking. There's a lot of risk in field banking in terms of these myrtle rust impacted species. They're constantly exposed to the disease and have to have sprays and management, but also they're exposed to the environment, weather risk, natural disasters, funding situations, and they do require a lot of space. They're actually absolutely essential, but they do require space. Seed banking is a perfect option for plants that can be banked in that way in terms of capturing the genetic diversity, but it's not possible for some of our plant species, and especially a lot of our tropical and subtropical species that produce recalcitrant seeds where they can't actually survive seed banking conditions. And it's also not possible for species where they're not valuable as a seed stock. So, for example, horticultural crops where you want to maintain the genetic integrity and where the seed would be outcrossed. Or potentially like the macadamia situation where you want to conserve one of the species but it outcrosses so much with the others that the seed don't have a pure species. So what we're doing now is using cryopreservation and tissue culture as a means to actually conserve these species ex situ. So this is just a showcase of what plant tissue culture can do. This was work of one of our PhD students. This isn't a native plant, this is avocados, but from one single bud we can get up to 500 plants out back into the nursery. And so it's a mass multiplication tool using plant stem cells to really amplify a particular genotype of interest. But also if we can start with a diversity set or a core diversity set informed by genomics, we can then help to amplify those plants so they can be useful for potential breeding programs or reintroduction programs. In terms of using tissue culture for conservation, there's a trade-off between the labour intensity, the labour and maintenance requirements and the storage duration. So bringing things into tissue culture is an essential first step for slow growth tissue culture and cryopreservation, but it does require a lot of maintenance, a lot of research and development, and plants have to be maintained in subcultures every single month onto new media. So what we're trying to do is transition our tissue cultures either into slow growth storage where you don't have to transplant them every month. You can increase that for a few months or 6 months, but also cryopreservation. And this is where we can actually store living tissues in ultra low temperature, so in cryobanks in liquid nitrogen. And that way we can store them indefinitely, just like Han here, and revive them again. And it's been used for a long time, actually. There are cryobanks around the world, especially for a lot of our horticultural root and tuber crops that are vegetatively propagated. But we have a huge gap in native species and in our horticultural subtropical species where the research just hasn't been done. There's a lot of benefits. Obviously, it's the only method for seedless or recalcitrant seeded species for really long-term space-effective conservation, ex situ conservation. It's space-efficient, it's lower risk than your field banking options. It's also true to type, so you know that what you're putting in is true to the genetics of what you're putting in and what you're going to get back out, hopefully. It's a long-term maintenance, low-maintenance option, and what you're producing that's coming out is disease-free because it's in a sterile tissue culture environment. So what we see is cryopreservation, which offers the long-term space-effective disease-free insurance solution for a lot of our imperiled Myrtaceae, but also other recalcitrant plant species. And so what I want to talk about today is this work. As Bob mentioned, we're running on short-term, competitively funded grant schemes, which is making it hard, but we're fortunate to make connections with these amazing partners, including the ANPC, the Royal Botanic Gardens in Sydney and Curtin University, and a bunch of these stakeholders who we could put together a proposal for 4 years to start to get these plants into tissue culture and develop the cryopreservation protocols. And this is actually led by Ricardo Mancera here out of Curtin University. And the aims of this is really to develop the first tissue culture micropropagation systems for some of these species. We've targeted 6 genera, Lenwebia, Gossia, Backhausia, Rhodamnia, Rhodomyrtus, and Syzygium. We also are trying to develop protocols for direct wild initiation, which is a massive challenge. Usually what we have to do is have plants that we maintain in a glasshouse where we can really control pathogen load and make sure they're beautiful and flushing a lot and we can get good material to bring into tissue culture. Wild collection is desperately needed, but also extremely difficult because of the pathogen load in the wild, not just metal rust, but lots of fungi and bacteria that you find on all surfaces. We're also developing the cryopreservation methods. Once we get them in tissue culture, we can do the cryopreservation development. And as a part of it, we've got a student also looking at the contribution of leaf traits to potential susceptibility. And we have some plants sent to us from the Royal Botanic Gardens that they've screened genetically, but also for myrtle rust tolerance of Rhodamnia. And we're comparing, the exact same species, but tolerant versus very susceptible plants and looking at their leaf structure under electron microscopy and those kind of things. But today I'm just talking about the advances in the tissue culture and the cryo space. This is our amazing team. All the work I'm presenting today is being done by these people, especially our students, which is such a powerhouse. We're very fortunate to get a lot of interest in our team in learning these technologies and some excellent students coming through. And massive thanks as well to the plant bank guys, especially Karen and Bryn from Kings Park, who we can workshop ideas with all the time, but also Jeff and Rod and Leanne, who's from Logan City Council, who helps fund some of our work as well. So these are the species we're focusing on at UQ. Lenwebbia, we've got 6 successions that were sent from Royal Botanic Gardens. Gossia gonoclada, which is the focus species for Logan City Council area. It's only found in their area in a bit of Brisbane in a really restricted range, and they're very invested actually in its conservation, which is great to have that support from Council. Gossia fragrantissima, which we had a small starter grant on from Australian Flora Foundation, so it made sense to keep going with that work. Bacchausia citriodora, lemon myrtle. It's not the most priority species, but because we're also an agricultural institute, it's of interest due to its biocultural and horticultural importance. And also a sneaky Decospermum, which was given to us as a Gossia, and we since noticed it was the easiest one to tissue culture and was looking very different to all the others. So we did a little bit of sequencing and found it to be probably a Decospermum strucoilicum, although there's a bit of blurred line to whether it's humule or not. So the general process that we go through, we first have to initiate explants from the glasshouse, and we then work on a media that can massively multiply those explants, because we need a lot of plant material and a lot of time to develop cryopreservation protocols and do it statistically and properly and make sure it's working. So it's actually, a lot of the beginning stages is just bulking up our culture material. Then we need to induce roots on that material, because we want to also show full pipeline from initiation into culture, but also release back into the wild. Acclimatization, which is a challenge in itself, coming from a perfect environment out to the real world. And then field validation. We just want to make sure the plants are morphologically normal, but also that they can establish again if they're going to ever be used in any translocation protocols. And then what we do, once we have multiplying shoots in tissue culture, we can actually bring those tiny little shoot tips, cut them into just 1 millimeter segments, which contains the shoot apical meristem. And the most important thing, obviously, in freezing any living tissue is we've got to get rid of the water or somehow vitrify that water that's not going to expand and kill the cells. So there's a lot of actual research optimization using various cryoprotectant chemicals, dehydration chemicals, without killing the plant cell, but protecting it from deep freezing, putting it in liquid nitrogen, and then reviving it again and showing that we can recover those plants, grow them back into full plants, and then actually get them back out into the field. It's very complex. So in a tissue culture media, just like in the soil, there's a lot of components that you need to add. It's a huge amount of optimization, and there's also light, energy source, temperature conditions. Every species is very different. It has an ecological niche in tissue culture, just like it has in the real world. If you get it right, it looks like this beautiful green Decospermum. If you get it wrong, it looks like this horrible avocado, which is where we started with avocados. The challenge is there are no universal plant tissue culture protocols. It is a big challenge. It's hard to optimize them, and you do need a lot of research funding and materials to develop the protocols. So what I want to talk about briefly is that we focus on the plant ecophysiology. Where does it come from? Where does it evolve? What soils is it normally in? What do we know about this species to help choose the first kind of basal media that we're going to put it on? But then we're also using computational tools to minimize the amount of experiments we need to maximize what we can test with the fewest resources possible. This stuff goes over my head at stats, so don't ask me any questions on this. But we do this design expert thing, and we can test a lot of media at once, basically. So what are the key learnings for Myrtaceae? I'll just summarize the progress we've made. We've got Gossia now, Gossia fragrantissima, a lot of cultures all the way through. We got it out to field, and yes, it's in cryo. Hooray, awesome. Gossia gonoclada, this is lagging a bit behind. We started later on this one. It's been a lot slower growing, and we had a lot of trouble getting it to multiply, but now we've just got that happening, so the cryo and the rooting experiments are starting. Decospermum, yes, all the way to field. yet to start the cryo. Lenwebbia, in just 9 months, our student has managed to get almost 2,000 cultures of this species in there, which is awesome. And Citrodora is a massive challenge. It's a very difficult species, so that one's still going. Here's some progress in pictures. Here's Maddie here and Jing, who've done a lot of this work. Gossia fragrantissima in culture, coming through to nursery. And we're actually very fortunate to have a really awesome relationship with the land care group in the native range of this species. And they're just helping us to see how these things establish in the field on a private property and see how they go, and helping to maintain those and monitor those. The same for the Decaspermum, which is nice. Here's just Jing in the field. And the good news is, for the first time, we've managed to get up to 70 to 80% survival of our shoot tips after freezing in liquid nitrogen. So this is what they look like. They look big here, but this is only 1 millimetre sections of shoot tip. They go into a cryoprotection process, and then you can see down here, they're starting to resprout after we warm them back up again. Gossia gonoclada, here are the cultures. Some have started rooting, which is nice to see, because rooting can be a big bottleneck in woody plant species. And this is Ben, who's done an honours project this year and has applied for PhD to continue this work, which is awesome. They've just started doing the shoot tip dissections and showing that they survived the dissection process. Lenwebbia, a lot of accessions and a lot of cultures now, which is awesome to see. This is Van Ann's work. And she's just started to do some trial shoot tip dissections. This is the size that this is a 1 millimetre grid. You have to do it under a microscope. It takes a lot of time and concentration, but these are they starting to sprout again after she's put it back on the regrowth media. Lemon myrtle, we do have cultures in multiplication, but we're getting a lot of dieback leaf drop. So the media for this still has to be optimised before we can progress. One of the major learnings we have is that you really have to optimise the nutritional health of the cultures first. It's so critical before doing anything else, and it impacts the cryopreservation success. So do that first. That's really essential. And that's where we use our response surface methodologies. A case study with Gossia Jing here did 42 different media testing different micro and macro elements. I'm just going to rush through here because I'm running out of time. You can see the difference in different media, poor cultures versus some of the better ones, just by changing the small changes in nutritional composition. And then comparing a commercially available media to what the program spit out as the predicted best media, you can see a big difference in the health of those cultures. And how that correlates is we were getting no success on the normal commercially available media. As soon as we modify and optimize a few things, you're getting up to 80% cryopreservation success. And another thing we've been really happy to see is that the predicted best media for one of those species is also applicable so far to what we're doing with Lenwebbia. So comparing the commercial media to the predicted best for Gossia also gave a big increase in health and performance. So where do we want to go from here? We obviously want to optimize propagation and cryo from lab to field, and we're working with the plant bank that will actually implement our protocols, which is good. We want to publish a new suite of optimized media for Myrtaceae that can be used by other people in this in vitro community. Adopt the technology for the accessions we're working on to a core collection informed by genetics. And then also propagate any materials that the community wants as well for potential breeding or coming out of breeding programs for translocation, but also bringing more species and accessions into tissue culture if we can get funding to keep going. So yeah, our vision is to really provide that capacity for developing the protocols for long-term banking of our recalcitrant plant species in Australia, especially in Queensland, and it's also our important bush food and crop species. But the challenge that I want everyone to take home is there is no infrastructure in Queensland to bank these species. There's no seed bank, there's no cryo bank, and we have so many of the important and imperiled subtropical tropical species that we need this technology for. Like, we're really the centre of where this innovation needs to happen, but there's no way to actually implement it. We're working with Royal Botanic Garden and Kings Park, which have the facilities, but I think that's something we need to really push for, and I'm happy to talk to anyone and be involved in anything that can help get that momentum for Queensland. So, thank you.